Good morning, Brunalini. Good morning. Good morning. Okay, we are left with a very little part of the MHC. Good morning, Sunita. Of course, we have done the entire MHC. I'm just saying. Good morning, Priya. Good morning, Saima. Good morning, Sumaya. Yeah. So we were discussing about the differences between the class 1 and class 2 MHC. We understand immediately. We understand the regulations of MHC. Friends, please kindly don't forget the regulations. They are extremely important for the part B questions especially. Good morning. Later we started discussing about antigen processing and antigen presentation. I want every one of you quickly answer my question. Everyone, endogenous pathway can be shown by class 1 or class 2 MHC? Think and answer. Endogenous pathway can be shown by class 1 or class 2 MHC. Everyone. Endogenous pathway can be shown by. Forget most of the people. Okay. So good. Very good. Exogenous pathway can be shown by the class 2 MHC molecule. Forgot. And endogenous pathway can be shown by the class 1 MHC molecule. Please kindly don't get confused with that, friends. Remembering is very important. Good morning, Grishma. And friends, we have very clearly understand how does this particular pathway will occur. I hope every one of you recall. First, what we have done, we made the peptide to break into smaller amino acids. The reason behind that, all the smaller amino acids cannot be expressed on the peptide binding cleft. So that's why by using the ubiquitin molecule with the help of a proteasomal complex, good, we have break down the large peptide into a smaller peptides. Later, with the help of a very important protein, that is what protein, friends? A tap protein. With the help of tap protein, the small peptides will enter inside. If you all of you quickly recall, in the endoplasmic reticulum, we have discussed very clearly there are three major proteins. One we can call thapacin, one we can call calreticulin, and another one we can call as calnexin. Friends, these three proteins used to associate with this particular MHC molecule. The moment peptide enter, what did I tell to you, friends? The moment peptide enter, these three immediately get dissociate, and alpha chain and beta chain associated with each other, and the peptide will be expressed over there. This is a exogenous pathway. And friends, if you quickly recall this is the endogenous pathway. If you quickly recall the exogenous pathway, which can be mediated by the class 2 MHC molecule. Quickly try to understand. I have explained it to all of you very clearly. Listen to me carefully, friends. This is the class 2 MHC molecule, and this is what, friends? This is the peptide binding cleft. Is it? Yes. Friends, listen to me very carefully. This particular peptide binding cleft can be blocked. The peptide binding cleft can be blocked by what chain? We can call it as the invariant chain. Kindly do not get confused with this particular point. And every one of you listen to me, friends. Now the class 2 MHC and the invariant chain, they will be packed in the form of a vesicle, and they will get bud off, and they will get modified in the Golgi complex. In the Golgi complex, friends, this particular invariant chain which is blocking the peptide binding cleft of the class 2 MHC can be digested. How it will be digested? Friends, it will be this invariant chain completely get digested. But what I told you, friends, Out of 100% of the invariant chain, friends, 90% of the invariant chain get completely digested and 10% of the chain will be left out. I hope all of you remember what is the 10%. We can call it as a clip. CLIP, clip we will be calling it as. Now friends, now whenever the exogenous antigen enters inside, immediately what is going to happen, friends? This exogenous antigen can be replaced by, who is going to replace this exogenous antigen? This can be replaced by HLA-DM and HLA-DO. I'll show you, just look into this. Everyone pay attention. Look into this, friends. All of you look into the diagram carefully. If you observe it, friends, listen. This is the class 2 MHC and we all know very well this is what, friends, a peptide binding cleft. This peptide binding cleft can be completely blocked by what chain? Invariant chain. There is no doubt about it. Now friends, this particular invariant chain in the Golgi complex, it will be 90% digested, and all of you please kindly look into the diagram carefully. There is a little part of the invariant chain is left out, which we basically calling it as the, friends, clip. Now all of you listen, whenever the antigen entered here, all of you look into this. This is the antigenic peptide. Once the antigenic peptide entered, the clip has to be replaced by this particular antigenic peptide. All of you look into this. The clip is getting released, friends, and that clip place can be replaced by the antigenic peptide. Now the question is here, who is going to replace the clip molecule with the antigenic peptide? That's the question. That can be done by two type of class 2 MHC molecules. We can call it HLA-DM, HLA-DO. Don't get confused, friends. They are purely MHC molecules only. We will be generally calling them as non-classical class 2 MHC molecule. What we will be calling them as non-classical class 2 MHC molecule. They are the one which is basically replacing the clip with the antigenic peptide. So friends, this is what class 1 and class 2 MHC molecule, which you people has to remember compulsory. The reason, friends, many times the MHC molecules were asked in the exam, either for the part B question or for the part C question. It's very important to remember entire information very briefly. Okay, so the next concept what we are going to read is the complement system. Friends, same way like MHC molecules concept only, this particular complement system is also exclusively important for the part B, friends, as well as the part C also. You know, the complement pathway is really interesting, and I can tell you that 90% of the information you people has to remember. At the same time, you have to understand it completely. The more nicely you understand the concept, very easily you can answer the questions. Okay, now listen to me very carefully, friends. Listen to me for 20 minutes continuously without any interruption. You will get a very good concept. Listen to me. What is complement molecule? Now everyone understand where this particular complement molecules will be present, and do you think all the complement molecules will be active all the time? No. Friends, listen. Complement molecules means they are a series of some 20 proteins. Now the point is here, my friends, why do I am using the word series? The reason I am using the word series of some 20 proteins, because friends, they always act in a cascade pathway like our blood clotting factors. We know very well, friends, If one blood clotting factor get activated, immediately it activate the another blood clotting factor also. There is no doubt about it. Is it or not? Now what is the point I want you to remember? Similarly, friends, understand, if one complement molecule get activated, it will activate the second to the third to the fourth to the fifth, and so on. So what you have to understand, there are, listen to me carefully, there are 20 complement molecules are there in the serum, and they will be always inactive. Whenever there is a requirement, they will be active, friends, and they will be working in the cascade phenomena. Kindly do not get confused with this particular point at all. And the another most important point I want you to remember, friends, the complement components are designated by a letter called as a C, followed by the number. Friends, everyone listen to me carefully. I told you na, there are 20 complement proteins are there. Friends, among the 20 complement proteins, friends, the most important complement protein among all, that is the C3. Please kindly don't confuse. C3 is the most pivotal, most important complement molecule among all. Kindly do not get confused with this particular point. And the last and final point I want you to remember, friends, listen. When one complement molecule are associating with the another complement molecule, they will be associated with the help of a B part. An example I am talking about. There is a complement 3. There is a complement 4. Example I am talking about. When these two complement molecules want to be associated with each other, first complement molecules can be break down into smaller part A, larger part B. Always B will be the larger part. Same way, complement 4 also can be break down into A and larger part B. So what I want you to understand is, friends, whenever the two complement molecules are joining with each other, friends, they will be always joining with the help of the large part only. Always remember this particular point. But there is exception. There is exception. Exception with what? Exception with only complement 2 molecule. Only one exception. Complement 2 molecule also break down into A part and B part. But in the complement 2, friends, A will be the larger part, that only associated with the other. Please kindly do not get confused with this particular point. So let me tell you one example. Suppose example, friends, pay attention. Complement 4 molecule and complement 2 molecule has to be associated with each other. Now we know very well, complement 2 break down into A and B, and I already told you in the complement 2, A will be the larger part, and complement 4 also break down into A and B. Now who will associate with each other? Complement 4B associated with the C2, A part. Okay, so complement 4B associated with 2A. See, we can call it C2A. C will be the common, so that's why we don't use multiple times. In the complement 4, B part will be coming. In the complement 2, A part will be coming. Always remember that. And last and final point I want you to remember, when I indicate a bar like this, when I indicate a bar like this, every one of you keep it in your mind, which indicates that it is having enzymatic activity. Kindly do not get confused with this particular point. When I indicate a bar like this, which indicates that it is having enzymatic activity. Friends, don't get confused. It's a very easy and interesting concept. Okay, just wait for 20 minutes. I'll make sure that you will get a crystal clear idea. Pay attention, pay attention. Now you can ask me a question. Sir, why our body require a complement system? What is the ultimate goal of the complement system? Now everyone listen. These are all the major functions performed by the complement system. Let me explain you one by one, all of you. So the first and simple and effective function performed by the complement molecule, that is a lysis of the targeted cell. Now you see this. This is the targeted cell. What do you mean by the word targeted cell? Why we are calling this cell as a targeted cell? Imagine this is the cell which is infected by the virus. Correct na? That's why we will be calling it as, friends, a targeted cell. Now this targeted cells, friends, can be completely degraded. Who is going to degrade this completely? It can be completely degraded by the complement molecule. Everyone listen. Whenever the complement molecule associated with the targeted cell undergo the complete lysis or complete degradation. How it will undergo the lysis and degradation, we will read in the upcoming slides elaboratively. Next one, opsonization. We already discussed coating of the antigen. Instead of coating, you can also use the word tagging. Tagging of the antigen or coating of the antigen. Every one of you look into the diagram very, very carefully, friends. This is the phagocytic cells. Let's take an example, friends, macrophage. Let's take an example, dendritic cell. We all people know very well, friends, phagocytic cells, they are responsible for what? To do the process of phagocytosis. Now listen, imagine I am a macrophage. If I want to do the phagocytosis of bacteria, first I have to recognize that particular bacteria specifically. Listen to me carefully. I am a macrophage. If I want to do the process of phagocytosis, I have to recognize that particular bacteria or pathogen very easily. How does this particular recognition is possible? Everyone pay attention. That recognition is possible with the help of a complement molecule. All the students look into the picture very carefully. To the bacteria who are associated, complement molecule associated. Friends, everyone pay attention. On the phagocytic cell, there are certain receptors are located. You all people can see it very clearly. Of course, friends, what kind of receptors are located on the phagocytic cells? Listen to me carefully. FC receptors are located on the phagocytic cells. Don't get confused with that. Now, to that FC receptor, who will comes and attaches? The complement molecule which are associated with the bacteria. Friends, here please kindly don't get confused. Here, bacteria do not come and attaches to the FC receptor. No. Bacteria which is associated with the complement molecule, those complement molecule will come and associated with this particular FC receptors present on the phagocytic cell. By that, as a result, phagocytic cells can do the process of phagocytosis very easily and very effectively. Kindly do not get confused with that particular point, friends. Next one, activation of the inflammatory response. All the students look into the diagram very carefully. Say an example, this is a particular WBC cell. It has to enter into the area where the damage occur. Why it has to enter into the damaged area? We're gonna read it in the process called as inflammation very elaboratively. It has to enter into the area where the damage occur. So how it enter? That process only we can call extravasation process. What do you mean by the word extravasation? Coming out of the blood circulation and entering into the damaged area. That is what exactly we can call it as extravasation. Kindly do not get confused with this particular point. Friends, pay attention. Now you look into this, right? On that particular WBC cell, FC receptors are present. Now to this FC receptor, who will come and attach us? Obviously, friends, complement molecule will come and attach us. The moment complement molecule will come and attach us, complement molecule will help this particular cell to come out, which we can call extravasation. Where it is coming out? It is coming to the particular area where it can start the process of inflammation. And look into this degranulation. You can take any kind of a cell. Let's take an example of a basophil cell. Let's take an example of a mast cell. So friends, we read in the first class of immunology, there are abundant amount of granules are present in the basophil. There are abundant amount of granules are present in the mast cell. Listen my words very carefully. Whenever there is a breakdown of this particular granule, what happen? All the chemical mediators present in the granules will be released, like histamine, like heparin, like platelet activated factor. They will all initiate the process of inflammation. We know that. Now the point is here, how does this particular degranulation is occurring? How does this particular breaking is occurring? Of course, friend, it is occurring with the help of a complement molecule only. You all people can see that on the basophil and on the mast cell, there is a presence of the FC receptor. Correct, na? So friends, listen to me. To that FC receptor, complement molecule comes and attaches. The moment complement molecule comes and attaches over there, they immediately stimulate the process of degranulation. By that, they can show the inflammatory response. Not only for that, even for the clearance of immune complexes. Look into the picture carefully. What do I call it this as? I will be calling this as the antigen antibody complex. Friends, why I am calling it as antigen antibody complex? Everyone pay attention, friends. I am calling it as the antigen antibody complex. Look into the diagram very carefully. This is the antibody. Friends, understand, the blue color one you can observe now. The antibody is associated with what? Antigen. So I can call it as antigen antibody complex. Friends, to that antigen antibody complex, what is associated? Complement molecule associated. Now you can ask me a valid question. Sir, how complement molecule associated with an antibody? We're going to see it in the upcoming slides. This is an antibody. Now antibody is associated with an antigen. Immediately what is happening, friends? The FC receptors of an antibody is getting expressed. FC receptors of an antibody is getting activated. Now to this FC receptor, who will come and attach us? Complement molecule can come and attach us. Now this what do we can call the antigen antibody complement complex. Now everyone listen my statements very carefully. Whenever a particular antibody associated with the antigen and it is also associated with the complement system, this entire complex can be easily recognized by the phagocytic cells like macrophage or dendritic cell. How? Undoubtedly, friends, with the help of FC receptor only. And friends, kindly do not get confused here also. To the FC receptor, antibody never attaches. To the FC receptor, antigen never associate. To the FC receptor, only the complement molecule which is associated with the antigen and antibody, they will only associate. Kindly do not get confused with that particular point at all. Now friends, these are all the different functions performed by the complement system. Now from here I want you people to listen every single step very elaboratively. Trust me, if you miss one step, you can't understand the another step because they are all interconnected. Let me explain you elaboratively. There are three type of complement systems are there: classical, alternative, lectin. Lectin mediated complement system means the complement system which can be mediated by the sugar pathway. When I use a word classical complement pathway, every student has to remember there should be a requirement of antibody antigen complex compulsory. Without the presence of antibody, the complement pathway is not getting activated. That is why we will be calling it as the classical complement activation. And friends, what do you mean by the word alternative pathway? Alternative pathway means what, friends? Antibody independent pathway. No, 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 Priya. No, 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 dear. Nothing like that, Priya. FC receptor, your question is valid, dear. FC receptors are specific for the IgE in the mast cell. Okay. At the same time, FC receptors also present on the mast cell, to which complement molecule also can come and attaches. Because Priya, your doubt is good. Here, when the IgE will come and attaches, dear, IgE will come and attaches when the IgE cross-linked with the allergen. Correct, na? When IgE is cross-linked with the allergen, then it associated with the FC receptor and it will break down the granule. So what I am saying is, we can also break down the granules. We can also break down the granules by the complement molecule also. When the complement molecule come and attaches also, it will allow the process of degranulation. Okay. So I mean to say, to one FC receptor, many kind of molecules will come and attaches. Ultimate goal is what? The process of degranulation. But when when IgE will attaches, when complement system attaches, is it depending on a particular condition. When allergen come, obviously IgE will come. It is depending on a particular condition. Okay. Okay. Now friends, don't get confused. Just listen to me for 10 minutes correctly. The classical pathway, which is the most important pathway among all, antibody dependent pathway. Don't get confused. Just listen to me. It's a very, very, very easy one. Easy to understand. Classical pathway. One point is crystal clear. If the classical pathway wants to be started, there should be compulsory antibody and antigen has to be associated with each other. Then there is an expression of the FC receptor. To this FC receptor, complement molecules will be associated. Such a simple step it is. So what I mean to say, classical pathway begins with the formation of soluble antigen antibody complexes. Friends, understand every point. With the binding of antibody to antigen on a suitable target such as the bacterial cell, then only, friends, classical pathway is getting started. And we already discussed, my friends, while studying the antibodies very clearly, there are two major type of antibodies or immunoglobulins are involved in the complement system. One we can call IgM and another one we can call, friends, IgG. So friends, you all people know very well, IgM is the major antibody involved in the classical complement system when compared to the IgG. I am not saying that IgG is not involved in the complement system. IgG also better complement activator. There is no doubt about it. But when compared to the IgG, IgM is the most better complement activator. I have clearly told you to which portion of antibody the complement molecule attaches. FC portion of the antibody. Everyone listen my statement. What is the first complement system that is getting activated? C1. Friends, one statement every student has to remember. That is, what is the ultimate goal of the complement system? Ultimate goal. The ultimate goal of the complement system is formation of a MAC, membrane attack complex. Suppose this is the bacterial cell. So they make a big hole. This is only called as a membrane attack complex. You're gonna see it. So through this membrane attack complex, perforins, granzymes will enter and they will activate the caspases. Finally, they will destroy this particular cell. So keep it in your mind. Ultimate goal of the complement molecule is formation of a MAC complex. Just now I told you, friends, the complement system, listen to me carefully. The complement system is getting started with a first molecule named as a C1 molecule. Pay attention friends, the C1 molecule can be divided into C1Q, C1S and C1R. All of you please kindly look into the diagram very carefully. The C1Q is a very long molecule. Friends, pay attention. The C1Q having how many subunits friends? The C1Q having 6 subunits. All of you look into the diagram. There is a long stack and one head. 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 You can look into that diagram carefully. Like that, 6 stacks containing 6 heads. Very simple. Now everyone listen to me. Actually the heads will be arranged like this. You can see the picture. The head will be arranged like this. Now you know friends, C1R and C1S, you know what they will do? They will bring all the heads like this. Very simple arrangement. Originally heads will be arranged like this. C1R and C1S will bring them close together. You can see, no friends, the green color ones. C1R and C1S unit, they are bringing all the heads come close together. Now pay attention. Now listen. If you listen for 5 minutes, you will get a very, very good idea. This is an antibody. And this is which portion of antibody? FC portion of antibody. Now everyone listen my statements very carefully. Sir, why complement molecule is not associated with this antibody? The reason is very simple reason. The reason is the antibody is not associated with the antigen. I told you very clearly, if the antibody is not associated with the antigen, complement molecule never ever attaches. Kindly do not get confused with that particular point friends, and just listen to me carefully. Now, antibody associated with what? Antigen. The moment antibody associated with the antigen, which receptor is getting expressed friends? FC receptor is getting expressed. Whenever there is an expression of the FC receptor, immediately what is happening friends? The complement molecule is getting activated. Now friends, which complement molecule got activated? C1 complement molecule get activated. Now I am going to explain you everything elaboratively. Focus. I have told you one common statement. What is the common statement I told to all of you? All the complement system works in a cascade pathway. What do you mean by that? All are working, all are activating one to other. So first, which complement molecule activated? C1Q. C1Q can activate C1R and C1S. Now everyone listen. From here the complement system is getting started. Now the C1S activate the complement 4 also and complement 2 also. Now what is happening? Complement 4 can break down into A and B. Complement 2 also can break down into A and B. But I told you very clearly, in case of a complement 2 molecule, there is an exception. What is the exception? I told to all of you. From the complement 2, A part will come. And from the complement 4, like other complement system, B will come. Then C4B, 2A will be associated with each other. Every one of you please kindly concentrate on the picture carefully. So C1R and C1S, what they are doing friends? They are cleaving the complement 4 and they are cleaving the complement 2 into 4A, 4B, C2A and C2B. Now this is the C2A. Every student listen. Now C4B and C2A associated with each other. And I told you very clearly, right? When I indicate a bar, over the complement molecule, what I have told you that? I told you that it is having an enzymatic activity. Is it or not? Yes. So that is only we call friends C3 convertase. Now the C4B2A with a bar we can call the C3 convertase. Kindly do not get confused with that particular point at all. And I also given you a hint. What is the hint I have given you? C3 molecule is the most important molecule among the entire complement system. Now what do you call it? This is C4b2A. C4b2A only we will be calling it as the C3 convertase. Some students will get confused here. Don't get confused. C3 convertase is the name of the enzyme. And you can ask me a question, sir, what is the function? You have to listen to me very carefully here. What is the function of a C3 convertase? Friends, the ultimate function of a C3 convertase is break down the C3. That's only function. It is breaking down of what? It is breaking down C3 complement 3 molecule into complement 3A molecule and complement 3B molecule. Kindly do not get confused with this particular point. Listen my statements. C3 convertase enzyme breaking down complement 3 into complement 3B and 3A. Here what we are having? Here we are having C4B 2A. To this what we are adding? We are just adding the 3B. We no need to add the C once again. Why we have to add the C? We already taken the complement as the C common. So we will just add the 3B. Now what are all there, friends? Now C4B 2A 3B. Once again you all look into the diagram. I have indicated the C4B 2A 3B with a bar. Which indicates that it is having a enzymatic activity. Is it? What do you call it, that enzymatic activity? What do you call that enzyme? C5 convertase. Kindly do not get confused with this point. Now all the students listen. Now you know, friends, what the C5 convertase enzyme will do. Everyone listen my statements. C5 convertase will break down C5, C6, C7, C8. All can be breakdown and all can be associated with each other. So C5B, 6, 7, 8. Now you can look into this because friends, in every complement molecule what we have taken commonly, friends, undoubtedly we have taken B only commonly. Correct na? And friends, I have very clearly told to all of you what is our ultimate goal of the complement system. I have clearly mentioned that the ultimate goal is the MAC complex. This is also many times asked one of the statement in the part C question. If you want to form a active MAC complex, C5B678 is not sufficient. What will be the reason? You only look into the diagram. C5B67, they can form only a small pore, hardly, friends, 10 angstrom in diameter. Hardly, friends, 10 angstroms in diameter, very small hole. But friends, if you want to form a membrane attack complex and if you want to allow all the molecules to go inside, at least the pore should be, friends, 70 to 100 angstrom compulsory. Now what I want you people to listen to me here. Now this particular C5B678 can be associated with a C9 complement molecule. Friends, don't get confused with this. And this C9 complement we can call polymeric C9 or poly C9. So you can see the diagram very carefully. This particular poly C9 associated with each other, they can create a big hole. Now this is what exactly, friends, we will be calling it as the membrane attack complex, or shortly called as, friends, MAC complex. Kindly do not get confused with this particular point at all. See, now what happens after the membrane attack complex, we will read in the B and T cells. But anyway, let me tell you, once the membrane attack complex formed, ultimate goal is what? We want to destroy this particular cell. Now perforin enters inside, granzymes enter inside, they will activate the caspases. then caspases finally will destroy this particular cell. This is what happens when the membrane attack complex occurred. Okay, whomever did not understand, I am summarizing the entire thing once again in this particular single diagram. Everyone please pay attention and try to understand it very, very clearly. Look into this picture, friends, very carefully. First, whenever antibody and antigen associated with each other, which complement molecule is getting activated first? All the students listen. Complement 1 is getting activated. Complement 1Q, 1R, 1S. What is happening then? Then the complement 1S, it is breaking down the complement 4, complement 2. Complement 4 can break down into A and B. Complement 2 also can break down into A and B. Now, which are getting associated with each other? All the students look into the diagram. Complement 4B and 2A are associating with each other. Then friends, it is going to give rise to a complex which we generally call it as, friends, C3 convertase complex. Yes or no? Of course, yes. Now friends, listen. Now C3 convertase, what did I tell you? It is having enzymatic activity. So C3 convertase is, look into the diagram, breaking down the complement 3 into complement 3A, complement 3B. Now to this particular C4B2A, who will come and attach us, friends? 3B will come and attach us. Now this entire complex, what do you call, my friends? This entire complex we can call the C5 convertase complex. And friends, what's the function of a C5 convertase? It breaks down C5, C6, C7, 8. In all, friends, the B fragment only associate. Then C5, B6, 7, 8 will start forming the membrane attack complex, but that is a very less. It is just a 10 angstrom only. Kindly do not get confused with that. Now the C5B678 complex associated with the polymeric C9, then they can form complete membrane attack complex, which is of 70 to 100 angstrom. So friends, this is called the first type of a complement system, which we can call classical pathway. And major point every student has to keep it in your mind, that is, it is antibody dependent, because you only try to understand, without the presence of an antibody, do you think the complement molecule will be activated? Not at all. That's why we strictly call it as antibody dependent complement pathway only. We can call it as a classical pathway. Among all the type of pathways, classical pathway only most important pathway. Okay, now look into the next pathway. Next pathway we can call it as alternate pathway. And friends, the moment you heard the name of a alternate pathway, what is the common point has to strike in your mind? The common point has to strike in your mind is antibody independent pathway. Correct now, friends? Whether antibody is present or antibody is absent doesn't matter. The pathway is taking place without fail. Okay, now listen to me carefully. It's a very, very, very easy. And one more point every student has to remember. Common point. Among all the complement molecules, C3 is the most common. And what is the ultimate goal of the complement molecule? Do not get confused. Formation of a MAC. These points you should never forget, friends. Okay, so alternative pathway does not need for antigen antibody complexes for the process of initiation. Now I want you people to listen my statements very carefully, friends. The alternate pathway actually starting from the complement 3 molecule, there is no doubt about it. Now my question is here, how does the complement 3 molecule on its own start the complement system without any antibody? The question or the point you have to remember, complement 3 molecule will undergo the spontaneous hydrolysis. Friends, please kindly do not get confused with this particular statement at all. What, friends? complement 3 molecule undergo the spontaneous hydrolysis to give rise to the complement 3A molecule and to give rise to the complement 3B molecule. So the point I want you to remember is spontaneous hydrolysis. On its own, complement 3 is getting cleaved. Don't get confused, friends. I will explain it elaboratively. Pay attention. Don't get confused. I'll explain twice. What you have to remember in the alternate pathway, complement system is starting with the C3 molecule. Ultimate goal is the formation of a MAC complex. That is our ultimate goal. Now, what did I tell you? This is the complement 3. It is undergoing this spontaneous hydrolysis on its own. Then it is giving rise to complement 3A and it is giving rise to the complement 3B. All the students listen to me very carefully, friends. Now this complement 3B, friends, listen to me carefully, friends. This complement 3B can be treated with a factor B. Don't get confused, just listen to me. Factor B. Now how does this particular factor B can be cleaved? Don't confuse. Factor B can be cleaved by a factor D. Factor B is the one which is basically cleaving the factor B. See, factor D will cleave the factor B into two parts. Factor BA, factor BB, same. We can break down into BA and BB. Correct now, friends? Now you can see here, this is what complement 3B. In the complement 3B, from the factor B which is coming, the large part is coming. 3BBB. You can see here, very simple. And friends, you all people has to look into this. Above the C3BB, I have written a bar, which means that it is having an enzymatic activity. It is acting like a C3 convertase. Listen, I'll explain once again. And I want you to listen to me carefully. If this C3BBB complex wants to be stable, it should always associated with a molecule called as a propyridine molecule. So once again, whomever did not understand, focus. Alternate pathway will always starts with the complement 3 molecule. Complement 3 molecule undergo this spontaneous hydrolysis and it is giving rise to the complement 3A and complement 3B. Everyone keep it in your mind, friends. This particular complement 3B can be treated with the factor B. First of all, how the factor B can be break down? Factor B break down by a factor D into BA and BB. Very simple point it is. Now complement 3B associated with BB. Now this entire complex we will be calling it as, friends, C3 convertase. Now if this C3BB complex wants to become stable, it should compulsory require propyridine. A very simple point it is. Now C3 convertase, see, I told you now this is acting as the C3 convertase. Now you all people know that. What's the function of a C3 convertase? Breaking down the C3 only. Now you look into this. This C3 convertase break down the C3 into C3B and C3A. Now don't get confused. You can take small only. Now what is the complex? C3B, BB. To that what we are adding? We are adding another 3B. We no need to take C once again. So C3B, BB, which is our previous complex. To that just we added another 3B molecule. That's all. Now I have indicated with a bar. Now it is calling it as what? C5 convertase. Now we all people know that. C5 convertase break down the C5 into C5A, C5B, followed by 6, 7, 8, followed by the 9, poly 9. Ultimately they are going to form the membrane attack complex. Then perforin and granzymes enter inside, activate the caspases and will destruct that particular targeted cell. Now what's the unique statement you can see here? There is no requirement of antibody and antigen interaction. It is just starting spontaneously from the complement 3 molecule. That's why we will be calling it as the alternative pathway. Kindly do not get confused with this. There is one more pathway and the regulation is there. We will see in the tomorrow class. That is the lectin pathway. Okay. Is it clear, friends? I hope it's clear for everyone. Okay. See friends, undoubtedly I will take the class today, no doubt, but not 8:30. 8:30 I have another class, I forgot to inform. I'll take it 1:30 p.m. I have already scheduled. Okay. If you are not finding a time at 1:30 p.m., please attend, whomever can attend, but others try to watch the recorded without fail, because we are going to start the gastrulation. No, there is a little part of the cleavage is left. I'll finish that. We will start the gastrulation. Very important it is, don't miss. Okay. I hope it's clear for everyone. Okay, that's great. I'll see you at 1:30 p.m. Please try to attend the live only. If you don't find the time, watch the recorded without fail. Okay. I hope Sadhguru you are following all the recorded now. That's good. Okay. Okay, friends. That's all for now. I'll see you in the afternoon. Okay. Bye-bye. Thank you.